The human body contains 37.2 trillion cells, and each of those cells contains many millions of proteins. Proteins are made of amino acids that form polypeptide chains. Proteins serve many functions in the body. The 5 key roles of proteins are catalytic, structural, transport, regulatory, and protective. These proteins are the key molecular building blocks for every organism on Earth. Today, we will be examining part of the process that transforms the DNA in our cells into proteins. This process is called the central dogma of biology, and is comprised of 2 parts, transcription and translation. Today, we will be focusing on the second process, translation. During the first process, called transcription, a DNA sequence is converted into mRNA. The process of translation is taking this mRNA that is produced and translating it into proteins. We start our process with a strand of mRNA that is located in the cytoplasm of a cell. The mRNA is comprised of 4 nucleotides: adenine, guanine, cytosine, and uracil. The nucleotides are arranged in sets of 3. Each set of 3 is called a codon. So here we have a ribosome, a tidy particle made up of nucleic acids and proteins. Each ribosome has 2 subunits, a large one and a small one. In this case, blue corresponds to the large and purple to the small. These come together around the mRNA, sandwiching it in between the 2 units. This is the initiation stage. First, the small subunit of the ribosome attaches to the 5' cap of the mRNA and moves along the strand towards the 3' cap. Once the ribosome encounters a start codon, which is always the sequence AUG, the initiator tRNA with the corresponding anticodon binds. tRNA, or transfer RNA, is carrying the amino acid that corresponds to its anticodon sequence, in this case methionine. The tRNA acts as a bridge, matching a codon in an mRNA with the amino acid it codes for. Next, the large ribosomal subunit joins to form the initiation complex. This concludes the initiation stage. The next stage of the process is the elongation stage, where the polypeptide chain gets longer. The ribosome will move on to the next codon. It is important to note that the ribosome reads each codon separately, not overlapping. In this instance, it will read AUG and then CCA rather than UGC in between. Here, the tRNA leaves, leaving behind the methionine amino acid, which is the first amino acid in the chain. The next tRNA, with the corresponding anticodon, will bind to the matching codon, carrying the appropriate amino acid, in this case, proline. The proline forms a new peptide bond with methionine, forming the beginning of a polypeptide chain. The ribosome then moves on to the next codon, repeating the same process as before. This time, the codon sequence is different, and the tRNA brings the amino acid leucine, and the tRNA leaves as before, and the leucine attaches to the peptide chain. This process is repeated, going codon by codon, until the ribosome encounters a stop codon, which will be the sequence UAA, UAG, or UGA. In this case, we used UAG. This starts the termination stage. The stop codon is recognized by proteins called release factors. These proteins mess with the enzyme that normally forms peptide bonds, causing the chain to separate from the tRNA, and the newly made protein is released into the cytoplasm. At this point, the ribosome will detach from the mRNA and the process of translation is finished.